A PCR is a technique in molecular biology used to amplify small and targeted segments of DNA. It helps create millions of copies of a particular DNA segment. Kary Mullis developed PCR in 1983 and received the Nobel Prize in 1993. The principle of PCR is based on the enzymatic replication of the DNA. In PCR, a short segment of DNA is amplified using a primer. The primers are short sequences that provides a starting point for DNA synthesis. The DNA polymerase can add a nucleotide to the pre-existing 3' OH group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase. Components of PCR. 1. Thermocycler, PCR machine. It heats and cools reaction tubes to specific temperatures needed for each step of PCR. 2. Target DNA. This is the DNA to be amplified. It can be single-stranded DNA or cDNA. 3. Primers. They are short sequences of nucleotides, usually 18 to 25 BP long, that serve as starting point for the DNA synthesis. Their main function is to provide a free 3' OH group for DNA polymerase to begin adding complementary nucleotides to DNA. 2 primers must be designed for PCR, the forward primer and the reverse primer. 4. Taq polymerase. A Taq polymerase is used as it is thermostable and works at high temperatures. It comes from Thermus aquaticus, a heat-loving bacterium, and is stable at 95 degrees Celsius, which is a thermophile. 5. Deoxynucleotide triphosphates. These are the building blocks of DNA. They provide nucleotides to form new DNA strands. Their phosphate groups provide energy for the reaction. 6. Buffer system. Maintains optimal pH and ion condition. It contains magnesium chloride, MgCl2, which supplies Mg divalent cations as a cofactor for Taq polymerase. The Mg joins to nucleotides as a cofactor which is recognized by the polymerase enzyme. Polymerase chain reaction detailed process. The process relies on a thermostable enzyme called Taq DNA polymerase, which can withstand high temperatures used during the denaturation step. 1. Denaturation. A purpose: to separate the 2 strands of the double-stranded DNA. The reaction mixture is heated to 92 to 95 degrees Celsius. The high temperature breaks the hydrogen bonds between the complementary base pairs. This causes the dsDNA to melt. 2. Annealing. Purpose: to allow primers to bind anneal to their complementary sequences on the single-stranded DNA templates. The temperature is lowered to 50 to 65 degrees Celsius. Short DNA sequences called primers bind to the 3 foot ends of the target DNA region. This step is essential for providing a starting point for DNA synthesis. 3. Extension. Purpose: to synthesize the new DNA strand starting from the primers. The temperature is raised to 72 degrees Celsius. Taq DNA polymerase adds deoxynucleotides to the 3' end of each primer. DNA synthesis proceeds in the 5' to 3' direction, copying the template strand. This results in the formation of new complementary DNA strands. Repetition of cycles.
Dénaturation, annealing, extension, make one cycle.
This cycle is repeated 25 to 35 times, leading to exponential amplification of the target DNA sequence, meaning the DNA doubles after each cycle. The principle of PCR is based on the enzymatic replication of the DNA. In PCR, a short segment of DNA is amplified using a primer. The primers are short sequences that provides a starting point for DNA synthesis. The DNA polymerase can add a nucleotide to the pre-existing 3' OH group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase.